Hello everyone. Welcome to our Ant Biology webinar series. We have fortnightly lectures on Saturdays and Sundays. We are very much delighted to have our speaker, Professor Sohan Modak, who has already given a wonderful lecture in our platform. Thank you again, sir. I am Shahali Vishwas, today's moderator. And to our audiences, you can post your question in the comment sections below. Continue till the end to know more about the work the speaker is going to say. Hope you will enjoy the talk. And feedback links will be given at the end of the talk. So stay tuned. Unfortunately, our moderator and convener, Professor Inna Rai Banerjee, could not join today for this for giving welcome note for the speaker as she is unwell. So, on her behalf, I am going to introduce Professor Sohan Modak to our audiences. Sohan Modak, BSc, is an embryologist and evolutionary bioinformation. He has doctorate from the University of Geneva and he did postdoctoral work at Oak Ridge National Laboratory and University of Kentucky. Lexington. He served as staff scientist at the Swiss Institute for Experimental Cancer Research, visiting professor at Ohio State University, and scientist engineer at French Center for Nuclear Energy, Grenoble. In 1973, he joined the University of Pune as professor. He founded the biotechnology training program NCCS Bioinformatics TIS. Professor Modak was emeritus at the Karnataka University, Harvard, and G. Ramachandran Senior Research Fellow at the IGIB Delhi. He published research in developmental neurobiology, molecular biology, and genomics. He now mentors students on molecular evolution. After his retirement as a head of the Department of Zoology from University of Pune, where he studied embryology and also conceived and developed the Department of Biotechnology and National Facility for Animal Tissue and Cell Culture. He appointed as a GN Ramachandran Fellow at IGIB Delhi. He then continued his interest in research on classification and evolutionary tree building that utilized more than a single gene. His notable discoveries include the demonstration of cheek length degeneration during normal development, which has later been termed apoptosis, and molecular phylogeny in 3D. And now over to you, sir. You can start your talk. The platform is yours.
Thank you very much, Shaili.
Welcome,
Good
sir.
evening, everyone, and it's a pleasure to address this presentation about molecular evolution. Actually, I call it Darwin's dream, because when Darwin worked on his during his voyage, he studied large number of wide variety of species of animals and plants in relation to their habitats also, and by examining he realized that there was some kind of relationship among different animals. Of course, the type of relationship was similar to what Linnaeus had said about taxonomy, but this was a different type of relationship in terms of evolution, that is the time-dependent generation of more advanced species. So this is what he was trying to do. So he examined a wide variety of fauna and flora, and he came to the conclusion that the evolution involves a serial improvement and serial augmentation in the uh, the structure and physiology of organism and their adaptations. And so he said the way to present this is by phylogenetic trees. Can you have the next slide, please? Shaili, can you give me the slide or should I do it myself? Okay. So Darwin therefore wrote this, uh, he built this small tree which shows how the mammals are positioned on the branch of the mammalia. And here you have this, which says that the affinities of all beings of the same class have sometimes been represented by a great tree. The green and budding twigs may represent existing species. Those produced with each forms with a succession of extinct species. The limbs divided into great branches, and these into lesser branches were themselves once were the free small living beings. Now this kind of connection of the former and the present buds by ramifying branches may well represent classification of the extinct and the living species in groups subordinate to other groups. From the growth of the tree, many branches are decayed and the new ones come into present, and they represent various orders, families, and genera, etc. The next slide. So Darwin's observation of animal and plant life recognized relationship between phenotypic variation and habitat associated adaptation along with ecological diversity. Darwin's analysis of morphological differences among species superposed the Linnaean taxonomic scheme and led to the concept of phylogeny in which the species were positioned on different branches at distances from the root and the stem based on the extent of their similarity among them. Now, at the time of when this work was done, very little was known about genetics, despite the fact that Mendel had already done the work on the basic genetic principles, but these were rediscovered subsequently. And so Mendel's observation established the genetic basis of phenotypic variation that allowed to compare related parameters quantitatively. Therefore, the method for constructing such trees involved assessing extent of similarity for a given trait or a character among a set of species. However, as each species being distinguished from differences among thousands of traits, multiple traits should be really compared for similarity to generate a consensus tree structure. Now, why this is being said is that nowadays we are looking at evolutionary trees based on molecular parameters. For example, we know the structure of DNA, we know that the contents of the genome. We especially have large number of proteins have been isolated and sequenced, and we know their composition. So one of the idea was to actually compare the molecular sequences. For example, in proteins are made up of a series linearly arranged string of amino acids. And so you take such protein polypeptide strings, and in the composition, the order in which the amino acids are organized on such a string, that you can compare among wide variety of species for the same protein. For example, in blood you have the globin, hemoglobin, and globin protein is present in all animals which have the blood. So the question is, can you take the blood from different species, isolate the particular globin protein, look at the sequence of amino acids of these proteins, compare them across the animal kingdom, for example, and see what kind of changes have taken place. And depending on the extent of similarity among different uh, different globin proteins of different species, you can actually build a tree. And so this is the kind of structure work that became very important during 70s and 80s when the amino acid sequences became available in increasing numbers. So let's look at the next slide, please. Okay. So the Darwinian phylogenetic tree was based on morphological traits, which is described by shape, size, position, and identity of each species. And we know that all of these traits are polygenic. Now, in the days of Darwin, of course, take example for a beak is a beak is a beak. So there are different types of beaks. So looking at a type of forms of the beak, one started looking at variation among different species of birds, and then start positioning them along some kind of phylogenetic system. Similarly, presence of tails, limbs, presence of scales or the hair, etc. So such traits were utilized to compare different species along and to understand their evolutionary relationship. Of course, now we know that each one of these traits is due to combination of maybe anywhere between 100 to maybe 500 different genes are involved in constituting a trait. And they may not be involved in a cluster at the same time, but to establish a trait, it's a process which takes cells to multiply, to undergo specialization, and during this process many genes come in, go away, the new ones express, and finally the particular character is formed. So what we say that such morphological traits are really polygenic. Now, in 70s and 80s, when the amino acid sequences became available and people started looking at evolutionary trees by comparing the sequences, trees were very interesting depending on the number of species from which the sequence of a given amino acid protein was available. But there was a problem with these trees because, for example, if you take the protein globin, alpha globin, and make a tree of such alpha globin sequences for a set of, say, 50 mammalian species, and you look at the structure of such a tree, which is the most primitive at the root, which is the most advanced outside, and then you take the same set of species, 50 species, and look for another protein, say lens crystallin, and you look and build a tree of that. Surprisingly, the tree for globin and tree for crystallin, the structure of the tree could not overlap. They didn't match at all, because it was very quickly realized that every gene undergoes random mutations, and when a gene X is affected, Y, Z, K, L, Y, N number of genes are not affected, while another gene may undergo mutation. So mutation is a random phenomenon affecting at different times different genes, and when it affects, it changes the amino acid sequence in the protein. And when the amino acid sequence changes, then as you compare the same protein amino acid sequence across many species, you start seeing that the sequences differ and they become increasingly dissimilar, or the extent of the similarity changes, decreases, and so that you can actually generate a tree structure from that. The problem was that the tree for crystallin and tree for globin didn't match. That means any tree, phylogenetic tree, built on the basis of one protein sequence doesn't tell you the story for all the entire organism or a polygenic situation. Because humans, for example, we have maybe as many as 20 to 40,000 genes. This number has never been really fixed. But nonetheless, that's a large number of genes, and roughly as many proteins are present. In case of Bacterium Escherichia coli, maybe there are 1,000, 1,200 genes. So number of genes have been increasing, therefore number of proteins have been increasing. Newer proteins are coming into being, and therefore to look at the evolution, we should have some kind of consensus understanding of different gene structures. Now let's look at the next slide, how this can be done. Okay, here is an example of structure of a tree, for example. Any tree, I call it phylogenetic tree. You can see at the bottom there is a root, there's a bifurcation blue root, then there's a stem, and there are branches one after another. So there are branches, and every time up from a point you get two branches, that particular point is a core, is a node, or there's an internal node, and then there is a terminal node at the end of the branch, where the branch finally ends and you get only flowers or final leaves or twigs coming. That's where it's called a terminal node. So you have terminal nodes and multiple internal nodes from the stem to the entire branch tree structure. So you have terminal nodes, and the terminal nodes are generally binary, 1 to 2. Sometimes you get terminal nodes which have 3 branches in them. And then you have got internal nodes. You have internal nodes which are terminal or primary, secondary, tertiary until you go to the stem. So this is a normal tree structure of the trees that we have in our garden or in the forest. The same principle holds true for phylogenetic tree in principle, and that's how we're going to look at it, how it does. The next slide. Okay, there are multiple ways to look at the trees and build the trees and look at different sequences, compare them to each other. And there are, for example, there are characteristic, there are things like, for example, you look at the how optimal when you start comparing two species for the presence of the same protein with whatever small variation in them, you first have to look at the optimality criterion. That is, how optimally comparable they are, what are the regions in it that are comparable, and which are the regions which are not comparable. So the ones which have the maximum similarity, they are optimally closer, and the others are optimally farther away. Now, there are a number of issues, so there are a number of criteria that are involved in doing this. There is also the same thing is one is based on the distances, another one is based on the characters. If you take a character, how close those characters are to each other in terms of similarity, or how distant they are. And there are so number of methods to actually look at it. And so different people build the evolutionary tree depending on different criteria. But finally, all trees, regardless of the criteria, should look very similar unless the criterion does not fit that particular nature of the tree. For example, the trees based on major morphological structures are not necessarily convenient for describing trees using the sequence analysis of, say, polypeptides or amino acid sequence in proteins. So one has to make a choice as the nature of the tree one wants to look at. The next slide. Anyway, these are technicalities. The important thing is that these are the different methods. For example, the one method is called maximum likelihood. Actually, it means how likely it is that particular set of the two individuals or entities, let's say, which are being compared, are going to be close to each other. So you don't want to compare an E. coli to a man, for example. There's a little likelihood that they will fall anywhere close to each other in a tree. Or there is a question of parsimony, or you look at what is the minimum number of criteria required to actually generate a tree structure in relationship diagram. Next slide. I will not necessarily describe these methodologies, but then there's another one is a neighbor joining. Like for example, if you have a branch and from branch there are sub branches coming, then which are the two sub branches which are closest to each other? And then if they further branch out, which are the ones which are closest to each other? So that kind of neighbor joining of the related trees structures are the ones that can be used, etc. So there are multiple methods and one can choose them depending on the type of the data you have and the type of the information that you're trying to compare. Now let's go to the next slide, which really tells better about. Now this is, for example, when you look at, therefore, what I'm really going to talk about today is how to build the trees using molecular data. Molecular data, for example, that is, if you have a sequence of amino acid of a protein called alpha globin extracted from hemoglobin, and such alpha globin from 300, 200, or 25, a number of species, then you can compare the sequence of the same protein from different species to each other. And comparison means the sequence always begins, all protein sequences, newly formed protein sequences begin with the first amino acid, which is called methionine. So what you do, you take the sequence of amino acids in the horizontal column, let's say this is the first species, horizontal column, you position the sequence of amino acid one after another. Then you take the second protein and put next to it in the next, and the sequence underneath, the third one underneath, and fourth, and so on. So let's say you have 50 proteins, so you have 50 lines going through, each line representing a sequence of amino acids. Okay. Now what you do, once you have that, then what you do, you start comparing. You say the first column is always methionine. Is the same true, is true for all the proteins? And the answer is yes. All proteins have the first column methionine. So when you compare them in a pairwise fashion, number 1 to 2, number 1 to 3, number 2 to 3, number 1 to 60, number 59 to 20, etc. All permutation combinations of comparison, position wise, column wise, and you get the following answer. Are they the same or they are not the same? If they are the same, the score is 1. If they are not the same, then the score is 0. Okay? So you get score 1 or 0 or 1, 0. And finally, if the size of the protein is known in terms of number of amino acids, then for looking at each column, you finally get a score for each pair. Each pair is being compared, that gives you a final score. And, and since if you have for each protein pair, and then you look for which pairs are closest, which scores are similar. So you say when the scores are very similar, they are probably closer to each other. Of course, the scores may be similar, but not necessarily that the protein sequence, amino acid sequence is similar, but at least the same number of residues are present. So in the first step, you actually found extent of similarity score-wise. So then you start making a table, who is number 1, number 2, number 3, number 4, etc. Then you again look at this table and say that there are these two guys where the scores are the same, but there are main differences in them. Oh, we have a problem.
Unfortunately, sir is having some problem, and I would request all the audience to have patience till sir comes. Have some patience. Our speaker will join us soon. Maybe he's having some problem. So yeah.
Sorry, we are facing some technical issues. Our moderator Shahili is trying to contact with Sir. So till then, you can comment among yourself, and we will join soon. Sorry for the technical glitch.
Alignment.
Okay. Sir has joined, and now.
The sequence alignment therefore allows you to find out how close one protein sequence is to the second one or the third. So if you have 50 proteins, sequences are available for the same protein, then you can ask the question, which two sequences are closest to each other? And if they are very close, that means they must have some kind of brother sister or some kind of relationship, child or parent kind of relationship. That means in the evolutionary scenario, they must be very close to each other, from where they must have evolved. So in this manner, you can take, say, 50 protein sequences, compare them with each other. What I mean is that same protein from 50 species really. Compare them with each other and then look at form the continuous tree. So you can then generate a dendrogram. For example, here on the right hand side you have a species number 1, and the species number 2 and number 3 are very close to each other. They are not identical, but there's a difference. Let's say this one has 100 amino acid, this one has also 100. Out of them, one is one difference in amino acid. This number 3 also has one difference in amino acid, but the amino acid which is different in number 3 is not the same as number 2. Therefore, from this node, you branched out to 2 proteins. So 2 and 3 are very close to each other. They are 99% close to each other. But there is 1% difference, and that is due to one different amino acid. Now then comes the next, number 4 is also different by one amino acid from number 2, but that means it has now 98 difference from number 1. And same thing, 5 has 98 difference from number 1. But if you compare now 4 and 5, that is 1, 2, 3, 4, there's a 4 amino acid differences and so on. So this is how you build a dendrogram or a tree branch. One branch giving rise to 2, then that giving rise to 2 more, or just continuing and so on. That's how the trees are built progressively. Okay, the next slide. All right. So there are multiple methods of actually building the trees. The original method was called Clistel W. I'm not really going to talk about it because it's available on the net, but it's very low accuracy and it's very complicated methodology for computation, and therefore it also tends to make large number of mistakes. But this is a very popular method because it's available online. All you have to do is give a sequence and set of sequences, and it'll give you back online tree structure. But this structure is not really very, very good. Yet there are a number of people use it because it's freely available online. And there are maybe at least 10 to 12 different types of, maybe more, methodologies for tree building. The two which are most accurate is the one which involves sequence alignment based on fast Fourier transform, which is a 3-step alignment, while another one is probabilistic consistency based method. And so these are very accurate methods, much more accurate than others. Anyway, one can choose the method depending on the type of tree you wish to build. So build a tree with multiple sequence alignment of polypeptide. Next one. Next slide, please. Here is a tree. For example, here is a tree. We found that there were 77 species of within the living kingdom, animals and plants and prokaryotes, etc. For the protein ATP synthase protein 8. The amino acid sequence for all 77 proteins were found. And so when they were compared with each other in the multiple sequence alignment, some of them very close to each other, they're shown by different color code because they actually turns out when not only they are close to each other, but they really constitute a very distinct part of the known animal or biological kingdom structure. Here these are all animals primarily and some bacteria that we have taken. There are no plants in this particular structure. You can see all the bacteria and fungi are located at the bottom. And they all this is the root of this tree, and there's a branching out. First branch goes to what are called prokaryotes, and next branch goes to eukaryotes. Eukaryotes are multicellular organism. Prokaryotes are unicellular organism and most primitive. So when you start looking at the tree, and suddenly comes a group of organisms which are very low level in the phylogeny and or taxonomy. These are invertebrates, very low level organization. Then progressively as they move into the very primitive kind of chordates, then the vertebrates, and then you go straight to the mammals and different other organisms like variety of different vertebrates or variety of different mammals that you can look at, and these are shown in the diagram. I will explain them exactly what happens. So right now I want you to look at simply this tree structure and concentrate on two boxes, this red box for mammals and the black box for lower invertebrates. And in between them there are 1, 2, 3, 4, 5 groups of organisms, red, black, green, black, okay, for example, position between them, and then there are 3 above and 1 below. So that is the general organization of that tree. We'll see the details later. The next slide done the similar tree for another protein for the same species, 77 species, cytochrome C oxidase. Now you look at this, here is this mammal structure, here is that other group. Now there is 1, 2, 3, 4, 5, and actually there are, this particular green one is really split. It's the same group of organism but split into 2 by intermission of some others. So 4 here. Now there are 3 groups above, there were 2 before. The next slide, the third tree. Okay, this is NADH ubiquinone oxidoreductase. Here is another tree. And now again the group of primates here, the lower invertebrates here. 1, 2, 3, 4, 5 again group. But look at the size of these constituents, and there are only 3 above, but they are much smaller because some of them have been split here into parts which have moved below. The point of the exercise is that if you now compare these trees next to each other and try to superposition, since they are for the same 77 species but for 3 different proteins, if they are really part of an evolution where all proteins evolve simultaneously and collectively together when they move from one species to another, then the trees should superpose completely branch by branch. In actual practice they don't, because every gene mutates independent of other gene. So if you're an organism which has 10,000 genes, the random mutation means you never know which gene is mutating or has mutated at what time, as a consequence resulting in the changing in the structure sequence of amino acid. The mutation will change the amino acid, therefore change the sequence of the polypeptide, therefore change the score for similarity, and therefore the position of that particular sequence will change. And this is what happens. So the trees never superimpose for a single molecular trait. Therefore we decided to build a consensus trees where we want to combine multiple traits together so that you can actually look at eventually polygenic consensus tree, which is really what Darwin was doing without knowing that actually every trait he was looking at was really composed of 300, 450 different gene structures, because existence of genes was not known then. It's only come later. So polygenic structures are the ones which are going to give a consensus solution. So let's look at the next structure. So this is what we decided to do, and this is what we call a Darwin's dream. A Darwin's dream is that if Darwin had known that actually there is a molecular composition which is really important behind every trait, every morphological trait is composed of products of many, many genes, then he would have dissected at molecular level each trait and then compared their collective differences for one species to another. And that would be Darwin's dream. So we said, let's look at Darwin's dream and make it ours. So we have developed a very powerful modular software tool to construct and visualize multi-parametric phylogenetic trees in a three-dimensional space depicting molecular evolution, evolutionary hierarchy, and species clustering. Remember, the trees that I've shown you so far, they're really two-dimensional trees. An actual tree, if you go in a garden, it's three-dimensional. You can go around a tree, you can go on the top of another building and look under to the tree from the top to bottom, or you can sit under the tree and look above. So that's what you need, a three-dimensional kind of positioning of parts of the tree. So as the branching pattern for a group of species is different for each trait, we have implemented a novel approach to construct a multi-parametric phylogenetic tree. Darwin's dream retrieves sequences from genes or RNA or polypeptide from online biological databases which actually have a collection of either amino acid sequence or nucleotide sequence, etc. So take this biological database, scan them for a particular protein or set of proteins, that how many species you have amino acid sequence of the same protein, and how many such proteins with multiple species which are similar to each other are available in the database. So you can take such traits and then we can then look at examine their amino acid sequences, compare them, measure the extent of similarity or dissimilarity, and then see whether we can actually plot this in a three-dimensional construct. So let's look at it now. Next slide, please. So this is the problem. Relationship among the same set of species in phylogenetic trees differ for different parameters. So solution. Position a group of species in 3D space based on similarity or dissimilarity distances for at least 3 parameters. Say, for example, 3 types of nucleotide sequences, or 3 types of amino acid sequences, or quantifiable biochemical parameters or biophysical parameters or morphometric traits. The point is, your traits must be measurable and quantifiable and compared on the quantitative fashion. And then when you do that, you get the extent of similarity among them. And then you can measure find a pair of species, for example, which are very close to each other, and you get another pair which within itself is close to each other, but the pair number 2 is away from pair number 1, and pair number 3 fits in between the number 1 and 3, and pair number 4 and 5 are farther away, etc. So all pairs distances should be obtainable, and then you can actually position them. And how to do that? So objective. Firstly, you do multiple sequence alignment in the picture that I showed you, and compute now their position in terms of extent of similarity. And then what you do, see, we were started with 3 polypeptides. The 3 pictures that I showed you, trees, each polypeptide we had the data for 77 species. We compared them with each other and similarity coefficient, and we build a tree. Now the numbers that we have which allow us to build a tree where who is the species at the root, who is the next one I'm mounting to the stem, and who is the farthest away, that particular order is based on the distance between number 1 and 2 and 2 and 3 and n. So that distance, similarity distance, can be then for one protein plotted on the x-axis, second protein on y-axis, and third one on the z-axis. And so you then get, actually in a three-dimensional space, you can position the species in between. Let's look at it, how it looks like. Next slide, please. Okay. For example, here you have a three-dimensional box. So what you do, you take a species number one for the protein. You say that protein was the one at the root. Whether it is or not doesn't matter, but you assume that it is at the root. And compared to that protein, in the sequence alignment score, you can position now with respect to the not protein, sorry, this is the species. Let's say this is the shark species. Protein number one for the shark, protein number two for the shark in the y-axis, and protein number three for the shark on the z-axis. So y, x, and z, three axes for the same protein. Let's say it is at 0, 0, 0 position, and with reference to that, you position those 8, 9 species which you have here. They are in the three-dimensional space. And now you measure the distance between this and species number 2, number 1 to 3, number 1 to 4, number 1, 5, etc. So now you get all pairs distances with reference to 3 proteins. Then you take another species, your shark you put in between. You take a mouse, for example, at 0 position. And now with respect to mouse, the same data is now plotted. By doing that, you get all pairs distances for each species pairs. If you have 9 species, you get 9 types of representation. And they all should give you the same pair distances, which they do. And this is called as the star distance. So now we have all pairs distances available. If you have 77 species, then you have got 77 times the species number 1, 2, 3, or 7 at 0, and all other 76 species within, you get the all pairs distances. So when these all pairs distances are available, you can actually measure these distances. Plus you have the distances available, and now with these distances you can actually build a tree because you already know what is the situation and how to do that. Here we have a three-dimensional plot, but we are always starting one species at 0. In actual practice, all the species should be within the box itself. So this method we have used simply to find the all pairs or each distance of each pair. Then we want to say, how do we build a tree from it? So here is the trick. The next slide, please. Okay, this is called as a multi-dimensional scaling. It simply is the following. Assumes you're sitting in a room, and the room, of course, is the volume, and there are little, say, butterflies are flying all over the place. Different butterflies, or say you have got sticks which are floating in this particular room, and they are floating at random, and once in a while they collide with each other. So depending on the volume of the space and number of the sticks, you can calculate the probability of collision of one stick to another stick and one type of stick to another type of stick. So then what happens is that, let's say in this picture you have some red stick, yellow, green, blue, black, and now when they start colliding, you can see, for example, here the green one has collided with blue one, but nothing happened. Then again, now the blue one has collided with the black one, and nothing really appeared to happen. At the same time, the red was colliding with green one. Finally, the blue and red, black and red collided, and the green collided. So first there was a joint here, then the third one, they form a triangle, then the fourth one collided with time here, and the collision occurs in the following way. Can I have the full screen? I will explain how this happens. Okay. Here, for example, here are two sticks. Okay. they are moving at random, and when they collide, let's say these two guys collide with each other and they form a joint. They form a joint. Okay, they form a joint. And this joint is now start moving at random in the three-dimensional place. Then comes another one which happens to collide with it, but the collision doesn't mean it will necessarily find a joint. It will form the joint only if it has the particular pairs that was already formed early in the all pairs distances. And when that happens, it will form a joint, so you get a structure like this. Then the fourth one will come, fifth one come, and finally all the collisions are finished. You will get actually a tree structure. Let's have the slide, please. Let's go back to the slide. Right. Okay, so as you go on, you start getting the structure formed. So you got one more collision. Here's now the fourth one is coming in. The fifth one has gone there. Sixth one is coming, seven, etc. And progressively you form. Initially looks like a network, and then it looks like a elongated tree structure or a spread tree structure. It may be like a tree. It may be creeper. It may be like a bush. Whichever, there are many types of tree structures, so any one of them can be formed. And you can actually, when the point is, when the collision occurs, the condition is that when the collision occurs, the collision must be same place. Let me explain. For example, here is a stick, a pair, A and B. Oh, I have actually written A and B on the red one, and there is a D and F on the yellow one. If A and B collides with D and F, nothing happens. But A and B collides with A and C, then A likes to join to A of the other one, so you'll form BAC kind of structure. Here, for example, you have A and C and B and D collide, nothing happened. Here they again collide. Now here you got that A and C and A and B, that A and B, they collided. Now they formed a structure. Then came in the third one along with it, which joined because now it has A joined, B and C are free. So there was a one which had end with B and A and C. They will form now a structure. Then the fourth one come in. So that's how the network and the tree structure is formed progressively. This is multidimensional scaling. This method, we were the first one used. It minimizes the stress function because it does the most probable combination based on the on the structure of the protein, and in this particular sequence similarity. The next slide, please. Okay, so when you do that, you go to next slide and we'll come back to this. Okay, when you do that, we are now the tree built based on the Euclidean distance. In the three-dimensional space is Euclidean space. And remember, if you go to in the three-dimensional space, and if you take 2 points, the vector between the 2 points is not a straight line in the three-dimensional space. It is a curve. That's what the discovery of Euclid, 3,000 years ago or something like that. So it's called Euclidean distance, is always a curvature distance. And so you get the Euclidean distance for 3 particular proteins. You can get it for 100 proteins. We have gone up to 15 so far. Anyway, that's irrelevant right now. The point is you can therefore build a Euclidean tree, and now what you have got again those 77 species, but now all the 3 proteins are together. So here there is 1, 2, 3, 4 groups. There were only 2 or 3 before. Here is that particular group of mammals, primates. There is a group here of lower invertebrates, 1, 2, 3, 4 clustered before. And now you can look at the composition of each one of them and the order in which they are positioned. Order because the first one comes from that particular branch. The next one comes from the branch later. So this particular node, binary node, gave rise to that one or that one. And from that one originated many species one after another. And in the fourth one there's another two, etc. So the most primitive one is the one which started the first, and the most advanced one that came the subsequent later. Now, by the way, this particular branch structure can be also shown inverted. It doesn't matter whether this is more advanced or that. Only the fact is that is the point of origin of the branch or the node which decides who is earlier and who is later. So there is this structure. Now what I'm going to do is I'm going to show you a slide where the order in which these guys are present in primates, in one protein, in the second protein, in the third protein, and in the Euclidean, the collective structure as it is. Okay, next slide, please. Okay, here this is how shown. For example, this protein, this particular structure has the orders as Papio, Hyloblatus, Pongo, Gorilla, Homo sapien, Pan, Paniscus or Troglodytes. Or here you have lower one in Lumbricus, Artemia, Albinaria, and Apis and insects. So insects and worms and that cluster group of organisms are here. Now the order in which they are present here and they are present here is something that can compare whether the order changes or not. So let's look at the next slide. Okay, or go back. Maybe you go back to this kind of structure. Go back. Here, for example, the first protein ATP synthase, which has this order. Then the cytochrome C, the same group of order, but look at first Papio, Pongo, Hyloblatus, Gorilla, Pan, Man, Pan. The second protein, Papio, first Hyloblatus changes place, Pongo come, Homo sapien is here, and Gorilla is between Homo sapien and the chimps. And you look at third protein, again Pong, etc. Now Gorilla is away, and again these three guys which form a cluster here are the same which form a cluster here. But every protein, the species is position is different. And when you do a consensus tree, then you get the collective picture of all the three, that you have again back to Homo sapien, Pan together. And this Gorilla is quite far away. And the same kind of structure you can get for the invertebrates. So you can actually show that the order in which the primitive or the most advanced species are located in that particular direction, that varies for every protein, because every protein undergoes mutation randomly at a different site or to the different extent. While you look at collective wisdom of all the three together, you get a more reasonable answer. The next slide. We can go back to that slide before. Okay, yeah. And now what we did, there is yet another method where before we started doing that, using this particular tree building method in the Euclidean space, some other people have already gotten the idea. We are not original in that sense, but they did it differently. What they did is the following. Let's say we have three proteins, which we have here, in which case I described. Let's say we could take the same three proteins, and is there another way to look at a consensus tree? And the answer is yes. And what you do is that, can you show me the screen? For myself, I want to make a demo. Siley. Okay. Here is a protein number 1. Is a protein number 1. Here's a protein number 2. So I join the protein number 1 to protein number 2. Whoops. Wait. Protein number 1 to protein number 2. and then I join the protein number 3 to it. Okay, this is called end-to-end joining sequence. So I take an amino acid sequence of protein number 1 and join the amino acid sequence of protein number 2 to the end. Normally what you do, protein has an amino terminal and carboxy terminal. So you take the carboxy terminal to which you join the amino terminal of the second protein, and to the carboxy terminal of the second protein you join the amino terminal of the third protein. This method is called end-to-end ligation. It was called, unfortunately, by people who think they're too smart, they call it concatenation. It's complete misnomer, doesn't matter, we'll stick to that terminology. This is a method of concatenating the proteins, basically. And now you compare the multiple sequence alignment of these three proteins together, and you make a sequence alignment column, one below another. Only now the length of the protein has changed. If a single protein is 200 amino acids, the second added say 150, third one added is 500, so 250 plus 150 plus 400. So your protein becomes very long, and longer it becomes, the all pairs distances become very difficult and complicated and time consuming to calculate, which is all right, but it can be computationally done. The only problem is the following. Say alpha globin of a species, let's say a fish, has a length of 500 amino acids. Alpha globin of a man has the length is 650 amino acids. I'm just taking, these numbers are not really correct necessarily, but I'm giving as an example. Now therefore then what will happen if I have alpha globin of man and the fish, I can, as long as the first amino acid methionine is aligned, the rest one it becomes a gap. I cannot align them anymore because there are no amino acids present. Now when I have therefore three sequences where each one in each sequence in each of these three string of three compared to the next string of three to the next, etc., the length of each collective string, three-word string being different, it becomes very complicated, very difficult to actually do truly a multiple sequence alignment. Anyway, as long as the protein numbers are small, it is okay. And such a tree always becomes a consensus tree to some extent. So we did that also for these three proteins. And let's look at what happens. Next slide. And we are almost now towards the end of it. No, that was the slide before, just one before. Okay, here we have looked at the Euclidean tree of the kind we already described, and now this is the concatenated tree for the same polypeptide. And look at now the red box primates: Papio, Hylobacter, Pongo, Gorilla, Pongo, Homo sapien, Paniscus troglodytes. Papio, Hylobacter same, Pongo same. Here is the Pongo different. Pongo and Pongo go together, gorilla is further away. Here the gorilla is split into two by splitting the Pongos. Otherwise, but most of the time this particular structure we're getting very similar kind of structure relationship. And this is more complicated in the invertebrates. Nonetheless, it's not very different. Now this problem is very simple, that as long as you have smaller number of polypeptides that you are going to concatenate, it's fine. They work as good as Euclidean tree, maybe sometimes even better, who knows. But as you increase the number of proteins, this problem becomes very difficult for concatenated tree. And we have compared that for 15 different proteins, which I'm not going to talk about, but the proteins with 15, the trees with 15 proteins are much more reliable than for the Euclidean structure than concatenate. Nonetheless, these are two different methods, comparable results, and they still give you a consensus structure. Next slide. Okay. Then it's possible to actually find out how good is your branch point. So there is a branch point. For example, I showed you. Remember that structure here, A and B, and this is the join point of A and B, which is called N1. That is node number one. This is binary structure, so that was the internal node. Then there's node number 2, N2, and C and D. So there are species A and B here, C and D here. And so the question is, can you really find out whether they are really next to each other, or should the A should be close to C more than B should be close to C, etc. So you can ask those questions, and there are softwares available. And we have now developed a software called All Pair Shortest Distance, which gives us the best results when it compares branch points to this tree. So we have far more reliable branch point analysis that we generate. The others aren't bad, but they are more partial towards one type of branching rather than other type of branching. Our method looks at all possible combinations. The next slide, please. And the next algorithm is called Fidelity Analysis. Now, here you have the problem. You remember I told you a tree built for one protein for 77 species compared to the 77 species tree for second protein or third protein, and you superpose them one over another, the pattern is not the same. And the question is, if the pattern is not the same, can you actually find out extent to which it differs from each other? That's one issue. And that means you can ask the question that in 2D phylogenetic trees, how faithful are the branch points? And that is called Fidelity Algorithm, which we published. We have developed this algorithm, and that algorithm does the following thing. You can look at the right hand side. For example, here you have 6 species: human, monkey, rat, rabbit, fish, and chick. And we have found that fish and chick find a clade, rat and rabbit finds another clade, monkey and human finds another clade. Monkey-human clade forms a subclade which joins to the rat and rabbit clade, and fish and chick clade is completely away. That's normally in taxonomy, this is what you expect, because the fish is the most primitive, then comes the chick, and then come the mammals, and among mammals, primates are close to each other, while the rat and rabbit are close to each other. This is expectation, and the question is whether this expectation is true, because there is an alternate proposal. We might actually find this structure. We might actually find, for a protein X, you might find this scenario. You get human monkey pair, and then you will find that rat is now showing a pair with fish, because rat protein has a sequence similar to the fish protein. And the rabbit is similar also to rat and rabbit, and chick is far away. So this variation will occur from one protein to another. And so if you want to look at the faithfulness or fidelity of this fidelity algorithm was developed, and that allows us to actually test every clade of the tree uniformly. The next slide, please. Now we are coming to the end. So here is the, therefore, method how to build a Darwin's gene tree. You take polypeptide database, like Swissprot, Uniprot, whatever it is. Find the species in which have the same protein amino acid sequence available for the set of species. Then download those sequences. The best database is Swissprot, because all protein sequences have been curated, vetted, and verified by experimentally, while other ones, some of them are computationally compared, but not really worked in the laboratory conditions and checked in the laboratory conditions. Anyway, you choose whichever you want, and you take the proteins and you do the multiple sequence alignment. I prefer probe cons or MAFT as the method. There are, as I said, there are many other alignment methods available. Then you compare now there are softwares available. There's got software called Phylip and Tree Puzzle, which allow you to build actually the tree. All you have to do is to give the sequence alignment data, and it will give you the tree. And it's online available, so you can provide the data online. They will provide you with a tree. Now up to this you get two-dimensional tree. But how to get multi-parametric 3D plots? There we have our own method. Remember I showed you how to compute star distances. Now star distances are Euclidean distances in 3D space. And then you do 2D phylogenetic tree for Euclidean distances. Then you take all pairs distance algorithm to find the shortest distance for each tree nodes. Then you do multidimensional scaling to get the sticks joined to each other end to end in different geometric configuration until you get all of them joined to get a tree structure. And now you do the fidelity assessment, and you can then animate the tree. Okay, let's go to the next slide. Unfortunately, now this is the tree for 77 species. It looks like a creeper, but it's actually a tree or a creeper if you want. Unfortunately, what has happened is that we had actually animated it in Studio Max. Unfortunately, this was done a number of years ago, and we have lost the particular stuff. So maybe we will next time we'll get it running. But let's look at the tree. It's very interesting. This tree says something else about what we thought so far about evolution. Okay, what is well established the following: the bacteria are here, the fungi come there, then the lower invertebrates come in here, more advanced invertebrates come in here, and then the intermediate ones, which are the protochordates, come somewhere here, and cyclostomes, protochordates followed by cyclostomes, cyclostomes are first chordates, the most primitive chordates, more advanced than protochordates. And then, surprise, the evolutionary tree showed in all books of evolution tell you the most primitive species among vertebrates are the cyclostomes, then come fishes, then come amphibia, then come reptile, and then from reptile you get two branches, one goes to mammal, another goes to birds. Lo and behold, this is different. What this is doing is the following thing. You're starting with the structure, ES structure, and you come here, protochordates, and now you come to the chordates. And from chordates, the first branching occurs. You got a branch here, which is going to the fishes and birds and amphibia. And second branch goes only to the mammals. This guy is elephant in between, so it's also a mammal. So here is a mammalian cluster which is originating independent of other three vertebrates, namely fishes, amphibia and reptiles, and four actually birds. They all come here. And so this is a very surprising discovery that the evolution is not really the way it was thought, but the branch point is somewhere near. This has been well tested, and we have now looked at similar kind of scenario with 130 species for 15 tRNA synthetases, and you get similar structure. Now, let's look at each part of this cluster, then you can see a little better. Unfortunately, I can't animate it here because I don't have that possibility, although I have done it many times before. So the next slide, please. Okay, here is, for example, the cluster where we are coming in the middle, right here, for example. Here is the Siona intestinalis. Here is the brown ticks, mosquitoes, honeybees. They are insects. All insects come in here. Locusta migratoria. There is the earthworm coming in between here. Then suddenly comes the protochordate, Balanoglossus. First comes the Balanoglossus, then comes the echinoderms, and then branch out to cephalochordates. and sacoglossus. And then comes the mixine. Now here comes the mixine is the hagfish, is a cyclostome, or petromyzon is more advanced cyclostome. And from the same cyclostome is very, very interesting. Petromyzon on one side and turtle on the other side. So they're very closely linked. Of course, this is based on 3 proteins only. And so therefore this is exactly what we find. But that doesn't mean this is the final picture of evolution. I will have to put in 100 different protein sequences to look at this to confirm, to get real. So 3 protein tree is consensus tree better than individual protein tree. But 15 protein tree will be much better consensus reach than 3 protein. 100 will be even better and 1000 will be even better. The next slide, please. That probably is the last slide. Okay, here is the other, now I'm going to show you the cluster of the lower vertebrates. Here you got chick here, coming the ostrich, then they're coming branching off here, and then come the fishes and frog in between. They're all intercalated. There's a rainbow trout here, there is look at the cyprius, gadfish, coelacanth. So all these structures coming in here. Okay. And look at this. Then you come to this branch. You got that elephant coming. This was that elephant coming here. Here is that elephant. The chick was here. Now you come in here and you have metatherians, and suddenly stripped branch point. Only primates here coming as a single branch point, and all other mammals are here in clustered. There is a cluster of mammals here. There are rodents form in different clusters. The bats form yet another cluster. The sea mammals form a different cluster. And so you got equus, for example, horses, etc., and rabbits, etc., and the rodents. And the base are the monotremes and marsupials. You got marsupials here and monotremes here. And then, so monotremes, marsupial at the base of both mammals, primates on one side and other orders on the other side. So this branch word point was never found or never looked for or never been described in any evolution tree so far. But it's based on only 3 proteins. Who knows, with more proteins added here, the consensus structure might change. Okay, next slide please. Okay, so general conclusion. We have developed a method to construct multiparametric trees. We developed algorithms to look at branch points and to benchmark discrete clusters against the taxonomic scheme. Now what you can do is that you can take the consensus tree, and for the same species you build a taxonomic tree. Taxonomic tree is based on linear taxonomy. And so now you are comparing the 2 tree structures, and you ask the question, are these 2 because here the taxonomic tree, you say this is a standard, gold standard. And how comparable to this gold standard is your phylogenetic tree. And therefore our algorithm APSDA actually compares this, and not APSDA, the taxonomic fidelity compares this and actually tells how good is your comparison. So we have done that, and so it's surprising. Ancestors, cyclostomes as ancestors, follow immediately. Vertebrates make 2 branches, one leading to mammals and other to remaining vertebrates. So it's not fish to frog to reptiles to bird and mammals, but fish, mammals, then from fish, amphibia, reptiles, and then birds. So amphibian lineage going to reptiles and birds is a different lineage unrelated to mammalian lineage according to this scheme as we have found it. As I said, tomorrow if I got 100 species, 100 proteins for the same species, the tree may look somewhat different. Anyway, the mammalian branch begins with marsupials, then splits into two sub-branches, one going straight to primates and others all other mammals. Again, another discovery. And in between, elephants have very unusual position in between. And so you can also allow detection of what is called as convergent evolution. So out of this, we have, when we compare then the different methodologies, different types of alignment protocols, and when we do actually fidelity points, we can actually find out, we find that single protein trees, they're about, total number of fidelity points expected are 18, and you get 14 to 15 points for single proteins. You get 16 to 18 points for Euclidean and concatenated tree. Maximum number of points is about 20. So you're getting better in terms of that. And that's about it. The next slide, please. I think this must be the last slide. Okay, these are the references. These are selected references. There are hundreds of people, hundreds of papers in this area. But these are the ones which are interesting in the context that I've described. Naylor-Brown is a classic method based on mitochondrial DNA, called it phylogeny, which actually compares the sequence of mitochondrial DNA. Densuk is one of the most interesting phenomenon which showed that tunicates and not the cephalochordates are closest living relatives to vertebrates. We have shown the otherwise, we do not agree with it. We find the tunicates actually way down the, for the three proteins that we looked at, that is to say, they way down, they compare with somewhere near the earthworm and things like that. While the cephalochordates, yes, they are very much at a position closest to living chordates. And this was the work on the, is published already, a review of what I have described today. Thank you. If you have any questions. Okay, Shaili, I finished.
Thank you, sir, for this wonderful lecture. I think audiences will be very much enriched with this talk. So, sir, we have two questions. Can we go through them?
Yeah, sure.
Okay, sir. Our first question, Shuranjana Halder, is fidelity analysis is little bit modified technique of UPGMA and cluster analysis?
Well, yes and no, because all fidelity analysis are similar to each other. Only the number of parameters you consider in one fidelity analysis may be different than another. But finally, it all comes to similar kind of cluster analysis. But the context, in that context, the nature of the cluster and number of elements on the cluster varies in fidelity analysis. Because UPGMA looks at all the clusters together. Here, fidelity analysis superimposes one pattern. So one is a pattern analysis. Ours is a pattern recognition analysis. UPGMA is looks at the pattern to build a tree. It is for building the tree. Ours is once given the trees, we compare them.
Okay, sir. Our next question is from Choitali Mari. For constructing a phylogenetic tree closest to the real evolutionary scenario, how shall we choose a set of proteins?
That's a very good question. And I would, in fact, what you would do is you will select the proteins. You will take some proteins, structural proteins. You will take some examples of enzymes. You will take some examples of carrier proteins. You can take some examples of the proteins which are actually signaling proteins, etc. So you may take representative examples of different proteins. So this is fine. The problem is that we do not necessarily find the sequences for them. And if you find them, you have to find the sequences of amino acids for one group of protein for a set of species. For the same set of species, you want to find the sequences of another group of protein, and the third and fourth and fifth. So people have done protein sequences helter skelter, in the sense as and when they wanted to look at it. And there has not been a particular scheme as to make a list of proteins to sequence and then sequence them. This has not been done. So when that data is available, this question can be answered. And I think it's a good question and needs to be answered. Right now we are looking at actually such structure. We have found about 25 species which have 5 types of different proteins available. So we are looking at that. Some of my students are looking at it. But other than that, 25 is a very small number nonetheless. It's a good beginning.
Okay. Thank you, sir, for explaining these questions. We have these two questions only.
Okay.
And now I will say that ma'am has, though ma'am couldn't join this session, she has sent good luck for this session, and is always very much thankful to you that you agreed to talk to this session. Thank you,
Thank
sir. Ma'am has sent this thank you note to
Thank,
you.
thank you very much. Right. I'm sorry I
And
went a little bit overboard in time. I took longer than expected, but at least I was tried to explain things in a manner it ought to be done. Thank you very much for the opportunity to talk to a group of people, and I hope they enjoyed it. And hope you
yes,
will
sir.
get some kind of feedback from them other than questions. Do let me know. If they have any
Yes, sir.
objections, I would like to know, especially objections. Right.
Yes, sir.
Or
Thank you for
suggestion to
this.
that matter. Thank you.
Okay, sir.
Okay, thank you. Yeah,
Okay.
yeah. Bye bye.
Okay, sir.
Bye, Shaili. Thank you.
Yes,
Thank you
sir.
for your
Thank you.
help. You have been very useful, very helpful in doing this construction, and I must thank you
Thank you.
primarily. Right. Thanks.
And now to our audiences, you can join our next session, which is tomorrow, on which will be given by Dr. Oishi Chakraborty. So stay until then. Stay tuned for that, and thank you. And you can also like and share these videos with your friends and colleagues, and obviously you can subscribe, and all the videos are archived in our channel. You can go through them, and you can obviously comment also, and till then, thank you and stay safe.